Greetings and welcome back. So we're in a series of vignettes that are dedicated to the idea of vectors, representations, collective coordinates, and all of that. And in the last few vignettes, what I did was to try to give an indication of how, by using the mass spring system, we could learn things about collective coordinates and dimensionality reduction. So we used that as an opportunity to create some data, the data being the positions of the two masses as a function of time. That becomes a time series. We sampled it, and we use that, in fact, to talk about PCA. So now what I want to do is I want to talk about a higher dimensional vector. That was a two-dimensional vector, x1, x2. Now we're going to talk about the high dimensional vector of the transcriptome, which is a 20,000 dimensional vector, let's say, the entries of which tell me the counts of each mRNA associated with each gene in the genome. As a preface to that, I first need to set us up for the idea of the cell as a bag of RNA, the transcriptome. So this vignette, which I think will be relatively brief, is about the more general topic of the cell as a bag of X. I love this idea. I know that some people find it to be silly in some sense. There's a whole history in the literature, and I'll refer to that in a little bit, about the cell as a bag of enzymes, and I think that that's being pejorative about it is missing the point. So as I say over here on the right-hand side, the notions of idealization, abstraction, and simplification, they are the lifeblood of science. They're actually even the lifeblood of the way we function in life. When I say the word red, I'm looking out the window, I see some red flowers. It'd be fun, if you were here, for you to tell me, actually, would you use a more refined word than red? They're actually not quite red. There's some pink in it, and so on. But we have this abstraction of redness. And as a first cut, it does the trick. Here, watch. Here's red. So you know what I mean when I say the word red. You can see it in your mind's eye. So we're going to abstract the cell, knowing full well that it is not a bag of genes, or that it is not a bag of proteins, or that the organism is not a bag of alleles. And yet, it's very convenient for us to adopt that fiction. So, the cell is a bag of X. That's our topic. I just want to say in passing, in case you decide to go examine our numbers, I still have my doubts about whether or not we got all of these numbers right for the fly. So, before talking about this, ultimately, in some sense, the starting point is experiments, measurements, more precisely. Measurements about the contents of cells, the contents of genomes, the contents of organisms. And the reason I'm showing this, I've shown it before multiple times in these vignettes, in my books, every talk that I have a chance to. I show this over and over and over again because I think it's a message that is not appreciated. This is from roughly 1911. It's from the book Atoms by Jean Perrin. It's nearly the last page of the book. And in it, he gives us on the order of 10 plus ways of measuring Avogadro's number. N divided by 22, Avogadro's number should be around 60, the value you see down here. And this is a bunch of different ways of measuring it. And to my mind, it's highly important that different ways of measuring the same thing square with each other. So as we talk through this idea of the cell as a bag of X, in the back of my mind, I will always have this idea that the cell is a collective of different types of macromolecules, for example, and we want to count them, and to do so is tricky. Okay, so the first question that I want to broach is the cell as a bag of genes. And so here we start with the exercise of the E. coli genome. I'm going to say that the length of the genome is something on the order of 4.6 times 10 to the 6th base pairs. And the question I ask myself is, how many genes are there in the E. coli genome? Our route to that answer is to say that the typical protein is on the order of something like few times 10 to the 2 amino acids. My evidence for that claim is shown here. You see 3 quite distinct organisms, and for my purposes, the typical protein size in all of them is the same, which is few times 100, a few hundred. So if that's true, then that means that I have an immediate gene length idea, which is few nucleotides per base pair, or per, sorry, I didn't say that right, per amino acid, times few times 10 to the 2 amino acids per protein, or per gene, maybe it's better to say it that way, per gene. So that gives me roughly 1,000 nucleotides per gene. And so the number of genes is equal to L genome divided by 1,000 nucleotides per gene, which gives us 4d600, I would say, as our rough estimate for E. coli. So here is the punchline of this thing, which is this graph, which originates, I think I originally learned of this or got it from Lynch, Mike Lynch, and maybe it's in his book on genome architecture, but I'm not quite sure. And I could have this wrong. I don't quite remember where I got it. But anyway, so this is also in Cell Biology by the Numbers, if you want to look at it there. But what I think is remarkable is that the slope here, this is for all sorts of bacteria and archaea, and this slope is basically 1,000 base pairs per gene. That's what you see, one gene per 1,000 base pairs. Obviously, this works quite well. So here's the comparison of the estimate. Yikes. Just bear with me. I don't know why it stopped sharing, but I'm coming back again. Just waiting. It's a little strange. Okay. So this is the comparison of the number of protein coding genes and the prediction of the number of genes. And what you can see is, for bacteria, these work very well. Our estimate works very well. It works terribly for eukaryotes. And an example is, so failure of this estimate is the human genome. And the way I would see that is, I would say, few times 10 to the 9th base pairs divided by 10 to the 3 base pairs per gene would lead to some sort of a wild prediction like few times 10 to the 6 genes, which is, as I say, wildly off. Now, there was a science article that you can read about by Elizabeth Panisi, I think is her name, and it was called The Gene Sweep. And what she was telling is that there was a bet in the scientific community about the number of genes that would be found in the human genome. And the winner of the bet was the person that guessed the lowest. So people had this prejudice that there were going to be more genes. So in other words, the philosophy of what I just did was used to some extent, obviously not exactly the way that I did it, because people knew about things like introns and so on. But I'm just trying to say that this estimate is spectacularly successful for prokaryotes and eukaryotes. It's spectacularly not successful for eukaryotes. That's something that we could go on a long discussion of, and I'm not going to. Okay, the next topic is the cell as a bag of proteins. So here, I just wanted to note that it's of interest to compute and to measure the proteome. So the proteome is the sum of all of the different proteins inside of a given organism. And what I'm showing you here is just a reminder of all the different ways that one might go about counting proteins. So I could use some sort of a readout, like an antibody or one of these two, or a luciferase or LAC-Z. I could do a fluorescent protein fusion. I could do the wild and very intriguing method of ribosome profiling. I could use mass spec. And my point is, note, that this is consistent with the spirit of this in the sense that here's all these different ways of measuring Avogadro's number. I'm proposing or noting that there's all these different ways of measuring and counting proteins. They're not entirely consistent. That's a very interesting discussion in its own right. But for our purposes today, I just want to point out, yes, people can go about that. One of the methodologies is mass spec, shown here. The notion is that you have some cell. In my case, I'm thinking of a bacterium. I break the bacterium open, and then I turn the bacterial proteins into fragments using an enzyme such as trypsin, which cuts them, and then run them in a mass spec. They get deflected based on their mass and on their charge. And so you get a spectrum like this. And in many ways analogous to the reconstructions that are necessary when you do shotgun sequencing of DNA, you have to do an assembly. You have to figure out what the fragments come from and then use that to count the overall number of proteins. And this is something that was done in a beautiful paper, one of my all-time favorites. It's by Schmidt and Heinemann and others. I think it maybe is in Nature Methods or something. I don't really remember. And maybe it's like 2018. They looked at 22 growth conditions. And here what we did is we tried to assemble all of that data. And you can see that the total protein copy number is something on the order of few times 10 to the 6th proteins per cell. This is as a function of growth rate. This is as a function of cell volume. We can make this estimate. Maybe we should real fast. I think I've done it already, but just for the sake of completeness, number of proteins per cell is the goal. And I'm going to do this for a bacterium. So that's the plan. And I'm going to go fast. But what are the things to know? The V cell is of order 1 micron cubed, which is the same as 1 femtoliter. The mass of the cell is, therefore, I'm multiplying the density of water times these things, 10 to the minus 12 grams, 10 to the minus 15 kilograms. So it's a 1 femtokilogram. The number of proteins then is, I'm going to say 15% of the mass of the bacterium is protein. So that's why I do 15 over 100. And I'm going to say that the typical protein is 30,000 grams per mole. And so this, when the dust settles, leaves us with few times 10 to the 6 proteins, which is entirely consistent with what I just showed you up here. Okay, take a look. Here's few. Few times 10 to the 6 proteins per cell. So the cell is a bag of proteins. Here, what I wanted to do is to talk about the organism as a bag of alleles. So there's this beautiful experiment that was done by Bury in the 1950s. It's a single author paper. You can go look it up. Gene frequency in small populations of mutant Drosophila. This was an experiment really about genetic drift. The concept was he started with 107 or so vials, each of which had 8 males and 8 females, all of which were heterozygotes. That means they had 1 red and 1 white allele. He then let them mate, and he randomly selected 8 males and 8 females in the next generation, and repeated this over on the order of 20 generations. And what he found is in the long time limit that the vials were either strictly red or strictly white, despite the fact that there was no selection. What does this have to do with the cell as a bag of X? This is really fun because we can do a calculation about this where what I do, and I think I already mentioned this, is I construct a vector. And this is number of guys with 0 red alleles, number with 0 red alleles. Oh man. Excuse me, I don't know why this keeps happening, but one of the things I learned from the pandemic era is we have to be flexible and adjust, and so I'm trying to do just that. So anyway, so this vector is, the first entry is the number of flies with 0 red alleles. 0 red alleles. And then this is the number of flies with 1 red allele. Sorry, how do I want to say this? I have 32 alleles total in my vial because there's 16 flies. There's 32 alleles. And what I mean here is the number of vials with 1 red allele in them. So number with 1 red allele, and so on. So this vector, which is 33 dimensional, so this is n32, this vector is it's an abstraction. It's basically pretending that these flies are nothing but a bag of alleles. I forget about the fact that there's a fly. I forget about the fact that there are wings. I forget about all of it. And I simply imagine that the population is an urn with red and white balls. That's it. This is something that Ernst Mayr referred to pejoratively as beanbag genetics. And as much as I admire Ernst Mayr and think that he was a superstar, I also think that he just never had anybody persuasively explain to him the power of a mathematical abstraction of a given problem like this, because this is a very useful entry point into the subject of population genetics. So our next vignette, the real reason for this vignette, the one I just did at all, was to set up the transcriptome. So we're getting ready to talk about representing the transcriptome, PCA, all the tools we use for the mass spring system. But in the context of the transcriptome, as a preface to that, my goal was to call attention to this very, very intense version of abstraction where we decide to call the cell or the organism a bag of X, where X is one particular thing that we intentionally focus on. So in the 50s, there was this idea of the cell as a bag of enzymes. If you look it up, this is actually from something that Hernan Garcia and I did for citations to bag of enzymes. And nowadays, this notion of the bag of RNA, I think, is a fun spin on thinking about transcriptomes. And so where we're going to go from here is I'm going to first talk about how to measure transcriptomes, and then we're really going to go into the details of how to mathematicize this large dimensional vector, and then how to find what it's trying to tell us using the tools of mathematics.